Yeah, welcome back students. In this video, I'll be discussing about the next assay, which is called as the subtractive and additive assay to build the cDNA library or RNA library. So, it's a subtractive and additive library. Additivity means adding some molecules to make sure that the whole total RNA is captured with specificity. So that is the subtractive and additive library. So subtracting means we have to have only specific type of RNAs removing, and we have to remove the rest of the RNAs which we are not interested with. So first thing is we have to probe or tag them, add some molecules and assay to make sure that specific type of RNA is present, and we have to remove the rest of the RNAs which we are not interested with. So this is the meaning of the subtractive and additive RNA, and we will build a library using the screened RNAs. That is referred to as subtractive and additive library. Is it clear? Okay, so have a detailed analysis of it, and I have given the notes in that. You can go through that, and you can also study any other book. So this is one of the new methodology, which is highly effective methodology for constructing cDNA libraries because it gives more specificity. Is it clear? So this method has been usually used to construct differentially expressed gene subtracted cDNA library from two cell lines with different metastatic phenotype. So slightly complicated, but remember, if we have two cells, one which may be caught with cancer, another one which is normal. Now the two expresses two different types of mRNAs with a slight differentiation. Okay, we don't know whether it is actually exactly differently expressed or not. So we can only know only when we do this assay. So or else easily when we take two different populations for evolutionary, for a genetic aspect, it's very easy. But when we take with respect to the cancers of the same cells displaying different, then it becomes little complicated. Got it? So what does this do? This technique characterizes two different mRNAs from the tissues, or you can say cells, which have the same function in two different organisms. Is that clear? Or sexes under normal conditions, or during different growth phases, after, or in case of researchers we use different treatments. So once we use different treatments, we can see how the same protein will be changing its same mRNA, which ultimately forms as a protein, will change its nature for every treatment. Is it clear? Or we will compare the two, normal and the diseased one, with a healthy, and find out what could be the difference. So subtractive hybridization actually detects the difference between different genomes or between cells where deletions or certain genomic rearrangement mutations as may have occurred. Is it clear? So you came to know the very important aspect of this subtractive additive genomic analysis. Is that clear? So here is the diagram. You can see very keenly, where you can see there are two means to analyze this. So slightly interesting what we have to do, where if you have two different types of arrangements in the RNA sequence, which we call them as, if we want to separate them, we use this methodology. So for this, two populations are required, which we mention it as tester. Another one is tracer, driver. So two different population, one is the tester, another one is the tracer, driver, which contains the target nucleic acid. So the DNA or RNA differentiates the one which has to be identified, which lacks the target sequence. The two populations, usually here two populations are hybridized with a ratio which is one is more and one is less. That is with respect to the driver and the tester. So driver will be always more than the tester. Remember that the driver should be always more than the tester. So more likely both will try to form an hybrids. So the driver tester hybrids are usually single stranded, and they come together and they will start to bind with each other. So for this, two important things we have to note. The driver tester will be forming one hybrid. Then the double stranded driver can form one hybrid, or any single stranded driver molecules are also formed, or if they are not attached, they will be removed. So like this, keeping only the driver and the tester, rest of the two types are removed. This is called as the subtractive step, leaving only the tester molecules enriched for sequences not found in the driver. Okay, enriched for the sequence not found in the driver. The same way, the driver tester, double stranded driver and any single stranded driver molecules. I will repeat it. The driver tester, double stranded, the driver tester, one type of molecule, the double stranded driver and any single stranded driver molecules, okay, are removed, leaving only the tester molecules, okay, which will be enriched, which will be accumulated more in number and will be not found in the driver. Okay, so for this, 5 steps are used. One is choosing the material, second one is producing the tester driver, producing the tester and driver, hybridizing them. So this is the most important difficult part, is producing the tester and the driver, then hybridizing them, removing the driver tester hybrids and excess driver is subtracted, and the remaining molecules will be collected. Got it? So this can be done usually in case of DNA as well as in case of RNA. Is it clear? So in case of, one more thing we have to note is, in case of this subtractive hybridization, the hybridization step is usually driven by excessive driver sequences. So the tester sequences that have complementary sequences in the driver population rapidly form driver tester hybrids, whereas the sequence unique to the tester population remains single stranded. Unique to the tester populations remains single stranded, or form tester pairs more lightly or lately. The ratio of driver to tester, the overall concentration of driver, the temperature and length of hybridization should be chosen based on the complexity of the driver and the tester, the abundance class of target nucleic acids, and the length of driver testers sequences are used to build the driver tester sequences. Okay, so what is the actual purpose of this subtraction step, which is very important, is to remove the driver tester hybrids formed during hybridization step and just keeping the testers more in number, leaving behind the tester enriched for identifying the target sequences. Is that clear? So the entire detailed procedure is again explained and given in the notes. Just kindly go through that and make a note of it. Is it clear? Okay, thank you.